Hello my dear students, this is Dr. 𝗲𝗵𝗿𝗺, your biochemistry faculty. Well, in this video we are going to discuss about the post-translational modifications of translation. So let's start up with the discussion. Post-translational modification. Now, what do we mean by this term? It means modification after translation, right? So if we talk about what is happening in translation, a polypeptide is produced. Now this polypeptide, examples are, you can say collagen is there, peptide hormones are there. So whatever polypeptide is produced, it has to undergo modification. Right. So this modification, once a polypeptide is produced, is known as post-translational modification. Very, very important. And if we talk about what are the examples of post-translational modification, the most important example is phosphorylation. Phosphorylation. Second is carboxylation. Right. Third is we can say that methylation. Fourth is hydroxylation. Then is ADP ribosylation. Right. And acetylation. There are others also, but we are just focusing on the major ones. Then one more is there along with this acetylation. We can simply say that ubiquitination. So these are the post-translational modifications: phosphorylation, carboxylation, methylation, hydroxylation, ADP ribosylation, acetylation and ubiquitination. Ubiquitination, or you can say ubiquitin protein is there. Right? Now, first of all, let's take the first one, phosphorylation. Now, if we talk about phosphorylation, it means there is attachment of a hydroxyl group, or you can say in that hydroxyl group we are attaching a phosphate. Okay. So what happens here is attachment of phosphate. Right. To make certain enzymes in a you can say to make the enzymes in a functionable state, or you can say whenever any active enzyme has to function, right, it will be converted into a phosphorylated derivative or a dephosphorylated derivative depending upon its function. So phosphorylation and dephosphorylation, these are the two very, very important examples. Just say if I quote the example of glucagon, and if I quote the example of insulin, what happens is in glucagon, the active form is with phosphate. So phosphorylation will be there, phospho, and it is phosphorylated. Just for example, if I talk about serine residue, this is serine residue. One is the active form and the other is the inactive form. So the difference is what? Difference is simply say if HH is there or OP is there. So this is we are changing the conformation of the enzyme. So this is we can say phosphorylated and this is dephosphorylated. This is one example I am quoting, and depending upon that different enzymes are there as per their function is there. So this is one. The second one is, the most important one is hydroxylation. Now hydroxylation is a very important post-translational modification which occurs on the molecule of collagen. What happens is, we know that collagen, which is synthesized in the body and it is the most abundant connective tissue. Okay. So it has repeats, and what repeats it has? It has glycine, proline and lysine as the repeats. So what happens is, once it is synthesized, whenever it has to perform a function, it needs to be hydroxylated. That means that this proline will be converted into hydroxylproline and lysine will be converted into hydroxylysine. And what we require for this? We require vitamin C, because vitamin C is acting as an antioxidant here, right, along with molecular oxygen. Okay, so proline and you can say heme is also required. So this is like a complex, or the enzyme here is prolyl hydroxylase and lysyl hydroxylase. So basically what is happening is, collagen which is being produced in the body, whenever it has to perform the function, it undergoes hydroxylation. So enzymes are prolyl hydroxylase and lysyl hydroxylase. And if we talk about this reaction, we all know this is a very important post-translational modification, because deficiency of vitamin C leads to scurvy. And we know that scurvy the main reason is that collagen formation is defective. So you can imagine the use of importance of this post-translational modification. Then another example is carboxylation. Now see, carboxylation of, you can say by vitamin K. Very important. We all know that vitamin K is very important, because vitamin K stands for coagulation. So what is the function catalyzed by this vitamin K? This is the only fat soluble vitamin acting as a coenzyme. And what is its function? It carboxylates, right? Carboxylates means it attaches carbon dioxide or COOH group to the glutamic acid present on the prothrombin active site. So it carboxylates glutamate present on prothrombin, right, or where it is the gamma-carboxylation. And what is its function? We know glutamic acid is already an acidic amino acid, and this will further add in more of COH group. So it will have 2 COH groups like this. So obviously if 2 COH groups are there, maximum adherence will be there with calcium, and overall prothrombin will be converted into thrombin and fibrinogen will be converted into fibrin and a clot will be produced. Fibrin, clot produced. This is also very important. So we can say that carboxylation is another very important post-translational modification of this prothrombin, right? Then fourth one is hydroxylation. We have discussed this. Carboxylation, methylation. Now what is this methylation? It is addition of methyl group, and this also helps in epigenetic modifications first of all. So I can simply write third, fourth, fifth. Methylation. Right? The function is, we can simply say that it is methylation is very helpful in epigenetic modification. Modifications. Right. Then after this, the sixth one is acetylation. Now if we talk about acetylation, this is a very important step. Actually what happens in acetylation, we know that nucleoproteins are there. What are nucleoproteins? The histones bind with DNA. Okay. And what are histones? Histones are simply the basic proteins rich in lysine and arginine. Now if we talk about the structure of DNA, we know that DNA is actually intertwined between the histones. Okay. So what happens is, these histones are like H2A, H2B, H3, H4 in a octamer like this. And this DNA is arranged like this. So this structure is known as nucleosome. So what is nucleosome? It is actually DNA with histones. Now what happens is acetylation and deacetylation, because these are the processes which are taking place simultaneously. So deacetylation also takes place in a sense that acetylation. So same, I mean like everything is going on, means removal is there, addition is there. Acetylation is addition of acetyl group, deacetylation is a removal of acetyl group. So if we talk about acetylation, so the enzyme is this histone acyl transferase, and if it is deacetylation, this histone deacyl acetylase. So this HAT enzyme will incorporate acetyl group. Okay, so it will increase transcription. Now how it will increase transcription? Because we know lysine and arginine are there and DNA is just wrapped like a nucleosome. So whenever DNA has to undergo the synthesis of RNA, it has to come out from the vicinity. Okay, so acetylation and deacetylation will help in this process, and that is why we say that histones are the most important elements for gene regulation. So if it is not required, lysine will keep them intact, but if it is required, lysine will be busy in this acetylation and automatically DNA will be exposed and it will simply undergo transcription following RNA, and RNA will undergo translation forming the proteins. So acetylation and deacetylation are very important processes. Similarly, phosphorylation, dephosphorylation are also very important processes. Whenever, because we have seen so many enzymes are nothing but the proteins. So many enzymes are active in their phosphorylated state, so many enzymes are active in their dephosphorylated state, like that. Then we come over to ubiquitation, and ubiquitin is simply, what is happening is whatever protein is there, once the function is over, it will be sent to a proteasome and it will be degraded completely. So I can say that once the function of a protein is over. Protein is over. It is sent for degradation. Right. So these are some of the very important post-translational modification. Most of the students get confused between translation and transcription. Post-transcriptional modification occurs after transcription and post-translational modifications occur after transcription, and post-translational modification will occur after translation. So depending upon the status of the enzyme, depending upon the protein which is synthesized, they are changed in their active, inactive forms and vice versa. So these are, and similarly if we talk about ADP ribosylation, it plays a very important role in cholera toxin. Plays a role in cholera toxin. Right. So this is all about the post-translational modifications. I hope this is clear to all of you. So this is all in this video. Thank you so much.